Hello, everybody. My name is Nilüfer Monhaseri, and in the following 20 minutes approximately, I'm going to present you our comprehensive solution for your regulatory T-cell research. Let's have a look at our agenda for today. First of all, I would like to give you a short introduction about regulatory T-cells. Then have a look at our Treg workflow in Miltenyi. Afterward, give an introduction or overview and translational workflow of Tregs, and at the end, summarize my talk and giving some key messages. Tregs are important subset of adaptive immune cells and control immune reactions for maintaining homeostasis in the body. In general, they have three main functions: suppression of overwhelming immune responses, which lead to the keeping of the balance and homeostasis in the body. The second is the suppression of immune responses against self-antigen and non-self-antigen, which protects against autoimmunity and mediate the tolerance in the body. And the last one is suppression of anti-cancer immune response, which is not really like the favorite functionality of Tregs because it supports the tumor growth. And Tregs have no unique surface marker. They are characterized by the combination of surface markers. In general, Treg cells express CD4 and CD25 and identified by the absence of marker CD127. And the naive Tregs can be identified by the additional markers like CD45RA. Moreover, FOXP3 is an intracellular marker of Treg, and expression of that is somehow a concept of the proof of the concept for the functionality of the Tregs. Based on their developmental origin, they're classified to naive, peripheral, and in vitro Tregs. Tregs has crucial roles in different types of diseases and provide novel therapeutic strategies for treatments of different immune disorders such as inflammatory disease, autoimmunity, transplant tolerance, and yeah, so on, and tumor immunity and so on. Okay, in the healthy situation, there is an immune balance between regulatory T cells and T effector cells. However, in some circumstances, this balance is disturbed, like GVHD, autoimmune disease, or in the condition of organ transplantation or rejection of the organ. And the reason which underlies the immune imbalance can be diverse. And adapter immune therapy with Treg cells lead to the reestablishment of the immune balance. And it's the reason that why Tregs are very attractive candidate for cellular therapy in the recent years. Just as a quick reminder that our company is a translational company, and regarding to Tregs, we provide you enough tools for very basic research to your clinical grade cell manufacturing. However, our topic for today is mainly on research workflow. Let's go through to the research Treg workflow step by step together. And as you can see, it's a complete work overview of our research grade Treg products, which cover the whole workflow for different species, such as human, mouse, and non-primate. Okay, let's start with the preparation of the sample. The secret of successful experiments lies in your starting material, because the quality of your sample preparation influence all downstream applications. Treg are very sensitive and fragile cells, as you might know, so be aware that you need to be very gentle and careful during the sample preparation. The main sources of Treg in mouse are spleen, lymph nodes, and as well as tumor, or it can be found in various tissues. In human, Treg can be isolated from PBMCs, blood, and different organs and tissue, and also tumors as well. So I would like to offer you one of the products or devices that many of you might know about it. It's a gentle max dissociator, in addition to dedicated tissue dissociation kit, which combining mechanical disruption and enzymatic digestion in the most efficient gentle way to obtain a viable single cell suspension from even hard tissue for downstream application or analysis. Our sample preparation products is not really limited to Gentle Max and our dissociation kits. We offer you Max tissue storage solution, Max smart strainers and pre-separation filters, and also dead cell removal kits. And here you can see an application example in which the skin tissue was dissociated with GentleMax protocol and GentleMax C-tubes. Afterward, for removing of clumps, the cell suspension was filtered using a 70 micrometer smart strainer. And at the end, cells were analyzed. Comparison between T-Rex yield out of manual and GentleMax protocol show the efficiency of dissociation by applying the GentleMax method, and it's really clear. And T-Rex CD4 FOXP3 positive cells has a higher number after dissociation with GentleMax. Let's go one step further and have a look at the isolation step. For isolation of Tregs, we offer you wide variety of products based on Max technology, which is a magnetic cell separation. And we have Treg isolation kits and microbeads, Max Express Treg isolation kit, Realis microbeads, and also you can benefit from our Treg cell sorter panel that I'm going to talk about each one more in details in the next slides. And accordingly, you can benefit from manual to automated cell separation system, as well as our unique cell sorter is it names is Max Quanti cell sorter. Let's start with the isolation kits. With Max isolation kit, you use our advanced cell separation Max technology, which works with minimal labeling with no effect on cell biology for full downstream capabilities. We offer you different Treg isolation kits for different species with various downstream application. And as you can see here, I put the numbers of purity approximately and the number of Tregs out of the kit exactly here for you, and you can have a look and see the quality. You just need to know for which application you're isolating your Tregs, and then you can choose the proper kit. For example, if you are looking for high purity cells for downstream in vitro separation assay, I would suggest you to use CD4 CD25 positive. Whereas if you need naive Tregs and you need to keep them long in culture, I would suggest you to use the CD4 positive CD49 negative isolation kits. And all of this application was tested in the company, and then we come up with this recommendation for you. Here, I would like to specifically mention the CD4, CD25 positive Treg isolation kit for mouse. This kit obey from the basic rule of our isolation kits, which is first depletion of the unwanted cells. Here is CD4 negative cells, and then positive selection of CD25 positive cells. And the final is really great. You will reach high purity and recovery. In addition, this kit is optimal for the researcher who wants to use the both fraction of the cells positive and negative as a two responders, CD4, CD25 positive as a Tregs, and CD4 positive, CD25 negative as T responder cells, which is mainly used for the in vitro separation assay, that it's important that you have the cells from the same donor. I hear from lots of you, especially who are working with mouse, that you have lots of different tissue at the same time in the lab, and automation is really demanded, and also handling several samples at the same time. I would like to suggest you fully automated system, Automax Pro Separator, which works easy, fast, convenient, and flexible regarding to analysis of several different sample at the same time. Okay, right now I would like to talk about Max Express Treg isolation kits, which is a unique kit for isolation of Treg directly from the blood. And the whole process takes 30 minutes. Max Express Treg isolation kit for human is the special kit which help you to isolate your Tregs directly from the blood. It is one of the most easy, fast, and efficient method, which gives the opportunity to you to save your time by skipping the density centrifugation step. And the next one is Realis microbeads. Realis microbeads help the scientists not only get a highly pure Treg, but non-labeled cells. It's really important. This revolutionary microbead technology allows to positively selecting the cells, but then removing the microbead and the antibody labeling to get completely label-free cells. And as the last isolation strategy, I would like to talk about sorting with max quant titocell sorter. Titocell sorter of Tregs is one of the approaches that I would like to emphasize on that a little bit because of two reasons. The first one is that Tregs are rare cells and very fragile. For isolation of ultra pure antigen specific Tregs regarding to some applications like CAR Tregs, and some of you need ultra pure Tregs for applications such as autoimmune disease models, or you have some organ transplantation models and so on. Tito can be a very good choice for you. And many of you are working on translational Treg project, which means that you are going to translate your research to the clinical application. It's good to know that we provide you all the consumable for Taito in two grades, research grades products and GMP grade products with extensive GMP documentation. It's a great opportunity which help you to move easily and smoothly from research lab to the clinical application. Let's have a look at an example. In this example, we decided to sort the memory Tregs. This panel of antibody you can see here we use. And just a point here, don't forget that we help you to design your panel. And also don't forget that all these antibodies are available in research grade and also GMP grades. Okay, we did the sorting. I know that the notes somehow are small, but I just wanted to show you the gating and so on strategy, that it's another point that we give you an extensive support. And yes, you can see the results. I just want to ask you to have a look at the numbers. As you can see, the purity are great, and reaching such a purity with healthy Tregs as a final yield is a fantastic results. Okay, after isolation of the Tregs, many of you might need to expand your cells. And for efficient culturing and expansion of your isolated Tregs, you need to consider three main factors. The first one is cell culture medium. Um, I just use the pointer here. We suggest you to use the TexMax, our dedicated T cell medium, which is free of animal-derived components and lots of other characteristics that you can benefit from that. The second one is the cytokines. The correct dosage, stability, and activity of cytokines are super important to have a good expansion of your target cells. For Tregs expansion, we offer you IL-2, and we have IL-2 in different grades, and I would like to ask you again to have a look at our webpage and get more info about it. And then the last but not the least, you need to have an activation reagent. We offer you two expansion kit that are efficient and available for mouse and humans. And unfortunately, because of lack of time, I don't have time to go through all these reagents and give you the example and showing the efficiency, but I would like to ask you to visit our webpage. We have other webinars ready to hear, use, and you can choose your best reagents for your research. Okay, the last but not the least step in Treg workflow is Treg analysis that I know lots of you have questioned in this part, but I'm going to explain you. But again, I should emphasize that just feel free to contact your local teams. We will support you. The analysis of Tregs can be done in each step of your workflow to check the efficiency of tissue dissociation, separation, or expansion. You can analyze your Tregs in two ways: phenotypic analysis or functional analysis. For phenotypic analysis, we offer you Treg detection kits and our recombinant antibodies, which name is reaffinity antibodies. And for functional analysis, we support you with the very exact and effective Treg suppression inspector kit for human. For phenotypical analysis of Treg, I would like to introduce our MaxQuant analyzer family of flow cytometry machines. Many of you know about them. It's a big family. We have different sort of MaxQuant analyzer. They are fully automated flow cytometers with 3 lasers and detection of up to 16 parameters, but it is not all. You need to really visit our webpage and ask our expert for further information. Beside a proper flow cytometry machine, you need to choose first high quality antibodies, and the second important point is to choosing the correct panels of antibody. So I would like to suggest you two solutions: either using the ready to use Treg detection kits, which are optimized antibody kits with pre-titrated antibody developed as a single one reagent solution available for human and mice, or if you need to analyze your cells broadly with several markers, I would like to suggest you to either visit our fellow cytometry application page because we support you with different panels of antibodies and the protocol, or contact us directly. We help you to design your proper panel and you can use the experience of our expert teams in this area. And regarding to reaffinity antibody, they are a recombinantly engineered antibody in which the FCR binding site was mutated to reduce a specific background, and highly purity and lot-to-lot consistency of these antibodies are great, and you can see in the application examples better. And let's have a look at our Treg functional analysis. I would recommend you to use Treg suppression inspector kit. What is it? This kit is the proof of the efficiency of your separation expansion steps because it shows you the functionality of your final Treg yield. In this kit, we have Treg suppression inspector, which is a polyclonal stimulus maxibits coupled to CD3, CD25, CD2. And you can see here when the responder T cell receive the stimulus here, they start to proliferate. But when you culture your responder T cells with Tregs after receiving the stimulation, Tregs suppress the proliferation of T responder cells. And in this way, you can see how functional are your final yield, final Tregs. Okay. And at the end, I would like to show you another application example on human CD4, CD25 positive regulatory T cells. Tregs were isolated from human blood using CD4, CD25 regulatory T cell isolation kits. After separation, the cells were analyzed by flow cytometer. Then the positive fraction as Tregs and negative fraction as T responder cells were used for the suppression assay. Just mention this point another time that you can use the Tregs and T responder cells from the same donor by using this kit, which is an important point for many of you. You can see in the results the absence of Tregs and presence of a simulation T responder cells here. T responder cells start to grow, and but in the presence of Tregs here, the growth of T responder cells is stops. It's the same situation in the absence of yes, a stimulation as well. Okay, now we are close to the end, and don't forget that Treg is a translational portfolio at Miltenyi Biotech and contains step-by-step solutions for your Treg workflow. And in summary, as you see, we cover the whole workflow of Tregs. We offer solutions for every pain points, and also we are more than just cell separation, because Miltenyi Biotech is really famous for the cell separation solutions, but we provide you different sort of products for sample preparation, expansion, and analysis, and so on. For further question, please don't hesitate to contact your local teams, and also our expertise, and send us emails. We will support you for sure. And regarding to Tregs, our webpage is really updated, and you can find all the resources there regarding to brochure, protocols, application notes, and so on. If you don't find your answer, again, contact us. And here, before we end, please acknowledge the displayed disclaimer. And at the end, I would like to say thank you so much for your attention